Hello, and welcome to my Week 2 presentation on microbial virulence. So what is virulence? According to the International Journal of Microbial Sciences, microbial virulence factors encompass a wide range of molecules produced by pathogenic microorganisms, enhancing their ability to evade their host defenses and cause disease. A little bit about the background on pathogens. As explained in the graph, pathogens start with host susceptibility, which is the organism that will feel the effects of the infectious disease that has traveled through the chain of infection, followed by modes of pathogen shedding, which includes respiratory tract, alimentary tract, urogenital tract, and blood, which are the most frequent sites of shedding. Diverse viruses may shed at virtually every single site. Next is reservoir host, which are populations, species, or ecological communities that drive disease dynamics because they can become infected by a disease agent and sustain transmission long enough to serve as a source of infection for uninfected hosts. There are 4 main routes of entry: inhalation, ingestion, injection, and absorption. Finally, there are modes of transmission, which include airborne, respiratory, formite, contact, transfusion, transcutaneous, enteric, and vertical. I have chosen to compare and contrast pathogens from the protozoa category and the fungi category. The first example is Giardia, which are microscopic single-celled organisms that are transmittable from human to human. For my second example, I have chosen ringworm, or tinea, which is contagious fungal infection caused by common mold-like parasites and is also transmittable from human to human. In these pictures, you can note the cycles of which Giardia and ringworm go through when infecting the host. Just as we discussed the background on pathogens, the chain of infection is very similar with 6 main stages: infectious agents, reservoirs, portals of exit, mode of transmission, portals of entry, and the susceptible host. Now we will compare and contrast Giardia and ringworm. So first we have Giardia, which is a protozoan parasite that infects the small intestines and adheres to the intestinal epithelial cells using a specialized structure called ventral adhesive disc. It attaches to the surface of the intestinal mucosa. It disrupts the host's absorption processes, and the symptoms include diarrhea and malabsorption. It evades the immune response and undergoes encystation, which is passed through feces and infects new hosts. It impairs the function of the gut and a fecal-oral route through contaminated water or food. Next, we will discuss ringworm. This is a fungal infection of the skin, hair, and nails. The fungi adhere to the host's keratinized tissues, where keratinases are used to break down keratin and facilitate attachment. It secretes enzymes and degrades keratin. It is confined to the epidermis, causing local inflammation leading to a ring-like rash, hence the word ringworm. Continuously feeding on keratin, the immune responds with localized inflammation. This is contracted through direct contact through humans, animals, and contaminated objects, and the spores can survive for extended periods of time in the environment. And lastly, the comparison between the two is that both evade the immune response, they are both pathogens, they are both transmittable from human to human contact, and they exit through direct contact. And lastly, we have my references slide. Thank you so much for listening to my presentation.